Hello, my name is Tori Slack, and my partner's name is Christian Schieffer, and we're going to be presenting on the drug Sugammadex under the brand name Bridion. So, what is Bridion? Bridion is a brand name of the drug Sugammadex. Sugammadex is a sugar-derived gamma-cyclodextrin and is the first selective agent for binding muscle relaxants. So, what is a gamma-cyclodextrin? A gamma-cyclodextrin is an octosaccharide derived from glucose, gamma meaning 8 glucose units. And what sets Sugammadex apart from other conventional methods of neuromuscular blockade reversal is the fact that it directly encapsulates and deactivates, making it much more efficient. To understand how Sugammadex works and why it's important, we need to understand how neuromuscular blocking agents work. And I am going to abbreviate those as NMBAs throughout the presentation. So how do they work? NMBAs are active at the nicotinic receptors on muscle cells. Nicotinic receptors are receptors that respond to the neurotransmitter acetylcholine, which is responsible for the contraction and retraction of muscle. And they are present throughout the central and peripheral nervous system, but more importantly, they are present on the muscle. And that's what we'll be focusing on today. So NMBAs displace acetylcholine from the receptor site and cause a relaxation response. And the NMBA we're going to be talking about today is going to be rocuronium. It's a non-depolarizing neuromuscular blocking agent. So that means it competes with acetylcholine for binding sites rather than depolarizing the ion channel that is a part of the nicotinic receptor. So no action potential can happen when acetylcholine is present on the binding site. It competes with the acetylcholine for binding sites. So to understand the conventional ways of reversal of these drugs, it's important to know that acetylcholinesterase is the drug that breaks acetylcholine down in the body. So conventional neuromuscular blockade reversal actually inhibits the acetylcholinesterase, which leads to spiked levels of acetylcholine. And then the neuromuscular blocking agents are replaced by the acetylcholine. So that's the conventional way. It's important to note that in these conventional ways, the neuromuscular blocking agents like rocuronium are not actually broken down in the body. They're just displaced and are still present in the synaptic cleft. And they are also not limited to the neuromuscular junction. And they actually act to spike acetylcholine levels in the muscarinic sites where acetylcholine is also active. So these cholinesterase inhibitors, they have some disadvantages and cause a multitude of postoperative issues, such as residual paralysis, nausea, and vomiting. The design shape of Sugammadex actually binds to non-depolarizing aminosteroids, in this case, rocuronium, and that's the neuromuscular blocking agent. So it does this because it has a cone shape, and it's due to the cyclic nature of the cyclodextrin as well as the abundance of hydroxyl groups on the secondary face, which pushes them outward to prevent steric hindrance while the thioether face is directed inward. So it creates this cone shape. In the picture here, it's actually kind of deceiving because the inner ring of primary alcohols is actually wider than the outer ring as shown in the picture. So it actually looks more like this. Basically, Sugammadex is a lot better than the conventional ways because it actually reverses neuromuscular blockade by encapsulating rocuronium within its core, and it reduces the risk of residual paralysis. And this happens in as little as 3 minutes since there is no residual rocuronium left, and it is removed through plasma by Sugammadex and is secreted out of the body through the urine. It all comes down to structure informs function, a really important chemistry point, probably the main thing that was ingrained in our brains. Basically, the shape and size consist of a primary carboxyl thioether face right here. This is the primary face around this cone. And then the wider secondary alcohol face. And both of these faces are hydrophilic. And then inside, we have a lipophilic core. The hydrophilic nature of Sugammadex is caused by the negatively charged thioether carboxyl linkages on the primary face and the negatively charged hydroxyl groups on the second place. And that's going to be really important. The thioether carboxyl linkages on the primary face also make the gamma cyclodextrin a lot more efficient in its encapsulation of rocuronium because there's a larger surface area over which thermodynamic interactions can occur. And it does increase water solubility as well. So the hydrophilicity and the resulting water solubility of the complex actually gives it the ability to dissolve in the plasma and be transported out of the body through the urine. The lipophilic inner core combined with this hydrophilicity can actually bind with lipophilic molecules, so it can take lipophilic molecules and move them out of the body through the urine. And in this case, rocuronium, the neuromuscular blocking agent that we want to remove, it is lipophilic, so we want to remove it out of the body. And rocuronium's shape fits perfectly to the Sugammadex size and length. So Sugammadex can surround it with its lipophilic cavity and bind to it, and it deactivates the neuromuscular blocking agent and can then carry it out because of the water solubility. So how does it perform in the body? The first of two steps is direct molecular encapsulation, and the second one is mass extraction. Sugammadex is actually administered intravenously and acts exclusively in the plasma of the body. So while in the plasma, direct molecular encapsulation happens as the carboxyl groups on the primary face of Sugammadex pull the steroidal rings of rocuronium into its lipophilic cavity, as shown in the figure right here. The steroidal rings refer to the typical core structure of a steroid, consisting of 3 cyclohexanes and 1 cyclopentane attached to each other. And this encapsulation process actually involves the quaternary nitrogen being in close proximity to the carboxyl groups on Sugammadex, and this increases the binding affinity of the compounds. So the rocuronium is now encapsulated into the plasma by the Sugammadex. The second step is mass extraction. So this happens when there's actually a concentration gradient created between the unbound rocuronium and the plasma, and this is in lower concentration. And then we have in higher concentration, the rocuronium bound to the nicotinic acetylcholine receptors. And the fact that we have this gradient actually pulls the rocuronium away from the receptors and into the plasma, and then they are rapidly encapsulated by Sugammadex. And I will pass it over to Christian to talk about the synthesis.
The synthetic pathway begins with a commercially available precursor gamma-cyclodextrin. Primary alcohols are substituted via the in situ production of a Vilsmeier-Hack reagent as a source of nucleophilic halides. Sodium methoxide is then added to make the reaction mixture slightly basic, around pH 9, before proceeding to the next step. Mercaptopropanoic acid and sodium hydride in DMF are added to form the thioether and the sodium carboxylate. Triphenylphosphine oxide was a major byproduct of the initial step, and because of its stability, it was found difficult to remove without affecting the product. Now we'll look at a proposed mechanism for this synthetic pathway. So we'll begin the mechanism by coordinating the halide to the triphenylphosphine. We then coordinate the carbonyl oxygen lone pairs to the phosphorus, form an oxonium ion. Nitrogen lone pairs then donate in, pushing electrons up to the oxygen, giving us the following resonance structure. We then have the iodine counterion attack the iminium carbon. Nitrogen lone pairs then come back in, breaking the carbon-oxygen bond, forming a phosphine oxide. The iodine pulls electrons from the triphenylphosphine oxide, giving us a stable byproduct, while also giving us the counter ion for our final Vilsmeier-Hack reagent. The next step in our synthesis is the halide substitution of the primary alcohols. We'll use the Vilsmeier-Hack reagent to coordinate the alcohol oxygen to the iminium carbon. This forms an oxonium ion, which greatly increases the rate at which the SN2 reaction takes place. While this is the final step in the halide substitution of the primary alcohol, I'll quickly show the regeneration of DMF in basic conditions that reduces solvent and reagent loss. Oxygen lone pairs will donate in, removing the iodide as a leaving group and reforming the carbonyl. The hydrogen is then removed by a base and we reform our original DMF. The final step of our synthesis pathway is the thioether formation in the presence of excess sodium hydride. We first deprotonate the carboxylic acid using the sodium hydride bond, as the pK of the carboxylic acid is about 5, whereas the thiol is only about 11. This forms hydrogen gas, pushing our reaction forward and forming our desired sodium carboxylate salt. The thiol is then deprotonated by another copy of sodium hydride, giving us the sodium thiolate as well. Sulfur, being less electronegative than oxygen, allows us to form the thioether over the formation of the ester. This is done through another SN2 reaction, displacing the iodine and giving us our desired final product. Since the introduction of Sugammadex, several parties have tried to create more efficient synthetic pathways. Newer developments focused on reducing byproducts, increasing yields, and decreasing the overall toxicity of reagents. Replacing the organic solvent DMF with DMSO increased the reaction time and decreased the reaction temperature needed. Penta and trihalide phosphines were used to replace triphenylphosphine oxide to reduce byproducts, but the toxicity of these reagents are much higher.